Hi, good afternoon, everyone. I'm Favour James from Obafemi Awolowo University, all the way from Nigeria. I'm very excited to be here. So today I'll be talking to you about text knowledge graph and how you can take it to convert a single paper into a Bell knowledge graph. I'll be discussing why we're using LLMs to extract knowledge graphs and how the code works and future directions for our project. So, knowledge graphs are very powerful tools for structuring and analyzing biological information because of their ability to actually intuitively represent data. However, constructing these knowledge graphs from unstructured scientific literature can be a challenge due to the high cost and the manual expertise that is required for manual curation. Traditional text mining methods can struggle with understanding context within sentences as sometimes they lack the background knowledge that is required to infer relationships in sentences that where the relationships are not directly spelled out. So large language models, which are LLMs, present an opportunity for us to overcome these challenges because they are trained on large, diverse biological literature. We can actually equip them with the ability to actually infer relationships in this kind of sentences. Existing ML projects tend to focus on extracting interactions from open access publications. However, with text knowledge graph, you have the opportunity to actually process and extract interactions from personal papers that have not been published, PDFs, and text. You also have the ability to share these knowledge graphs online through index. So here's an example where we process the sentence AKT2 also phosphorylated S326 of HSF1, but showed weak ability to activate HSF1. The statement in blue is the Bell statement. However, we noticed that there was some information loss where weak ability was not captured. So how does this tool work? Our tool has the ability to receive two different input mode, the PubMed Central ID or your file in the PDF or text format. So if you provide a PubMed Central ID, we extract the article's text from PubData. However, if you provide your paper in either a PDF or text format, we pass the paper using Microsoft Markdown model. Internally, this pipeline extracts paragraphs from the paper. Then these paragraphs are then annotated using Gilda, where Gilda identifies the entities in each of the paragraphs, then grounds these entities. Next, the paragraphs alongside the annotations are sent to the LLM, which is the OpenAI GPT-4o. The LLM then extracts interactions while depending on these annotations from the sentences. So the results we get from the LLM are Bell statements. Bell stands for biological expression language. So these statements are then processed into the CS2 format, which is our output format, and is a standardized JSON-based structure for Cytoscape. It captures nodes, edges of these interactions, so you can easily load or visualize these networks in either Cytoscape or Index. So once you get the CX2 file format as the output, you can choose to upload this network on Index. You can choose to analyze it locally on your system in Cytoscape. So our tool has two different usage modes. You can use it's available as a Python package, or you can use it inside Cytoscape Web. So so in order for the LLM to actually extract detailed interactions like we wanted, we ensured we gave it a very detailed prompt where at first we gave it a list of instructions where it's supposed to follow. At first, we told it is going to be acting as a biological expert, and his main task was to extract interactions from paragraphs. The next thing we did was to give it a list of entity namespaces that it must adhere to, such as HGNC, Go, and Kirby. We also mentioned to it that a sentence with less than two entities cannot have an interaction, because for you to have an interaction, you need at least two entities in a sentence. We also gave it the type of Bell interactions and Bell functions that exist. Another thing we did was to give a few short example outputs of the kind of Bell statements we expect it to give, and it also received the list of entities and notations that were done by Gilda. At first, actually, we tried allowing the LLM to extract interactions from papers without actually giving it the list of namespaces. However, we we realized that this caused the LLM to hallucinate identifiers for genes, whereby it was giving a wrong identifier to another gene and was mixing up gene names. So this is why we referred to giving it the list of namespaces. So here's an example network of the paper that was processed by our tool, and this is a zoomed in view where you can see the interaction that I showed in the former example. This is the AKT2 and then the HSF1 and the directly increased interaction. So as I said earlier, you can access our tool in Cytoscape Web, which is a tool for visualizing and analyzing networks. So we have a how-to section on our GitHub repo in case you want to make use of our tool in Cytoscape Web. This how-to section is going to guide you of how to make use of our tool on Cytoscape Web. So something that we did, we tried evaluating our tool and there was our results, and there were some challenges we faced. The first challenge that we faced when doing evaluation of knowledge graphs was that we noticed that knowledge graphs created in BEL are actually limited. Another thing is that existing interaction large databases tend to not map directly to sentences. So what do I mean by that? They map papers to interactions. So we don't have things that map sentences to interactions. And also we try to compare our tools against like large scale automatic extractions such as Indra. However, during the comparison, we realized that some of these interactions that were gotten from Indra were actually incorrect, and this actually limited our evaluation options. So we also have limited human reviewers with BEL experience. A recurring issue that we noticed was that multiple valid extractions are possible for the same sentence. So I don't know if you can see my example. It's very tiny, pardon me. So the first row is the example gotten from Indra. The second row is the example gotten from our tool. And these are very similar extractions. However, there's a difference which is marked in red between the extraction gotten from our tool and the one gotten from Indra. So another thing we did was to qualitatively analyze our responses, where we gave 5 different reviewers a sum of 50 BEL interactions for them to review based on 6 main error criteria, which included incorrect entities, interaction error, invalid relationship, identifier error, correct can improve. So the standard for a statement to be correct was the criteria all correct, which meant the statement did not have any issue with any of these errors. On average, we had about 28 all correct statements, excluding the reviewer 4 was a kind of outlier because the person's review did not align with most of the other reviewers' review. So we also noticed that the identifier errors were sometimes due to the Gilder entity extraction that was done for the statement. We also compared the outputs gotten from textKnowledgeGraph to the outputs gotten from Indra on the same textual evidences. In total, we passed 38 evidence tests. And for textKnowledgeGraph was able to extract interactions for the 38 of them. Indra was able to extract interactions for just 29 out of these 28. Our textKnowledgeGraph had a total of 67 BEL extracted statements from the 38 sentences, and Indra had a total of 29. So in order to kind of get a comparison of how they relate to each other, how they complement each other, we split each of the BEL statements into main components: subject, object, interaction type, object namespace, subject namespace, as you can see in the graph there. And we noticed that Indra and our tool seems to align on object namespace and subject namespace. However, they misaligned a lot on subjects. And this is an example below too that is very small. These statements were very similar. However, our tool added some things, some terms like the go term and the ACT, which Indra didn't add in his own interaction. So like I said earlier, we tried to get a comparison of how this result actually complemented each other, and we developed a kind of scoring logic to rate these components, whereby if they matched on relationship type, that statement had a score of 0.4. If they match on subject or object, the statement had a score of 0.3, and if they match on object namespace or subject namespace, the statement had a score of 0.25. And the best possible score was 1, where we had a statement that actually had 1 for the two tools, and the lowest possible score was 0, where they didn't match on anything. So out of the 29 overlapping sentences between Indra and LLM, Indra was able to match our LLM a bit on 17, and there was no match for 12 of them. So 17 had non-zero matching scores, and we had the average score of 0.63. So we also tried something where we used LLM to actually rate the semantic similarity between 29 overlapping Indra and text to knowledge graph statements because of sentences like the one below, where they can be very similar but they are different in structure. And we had three categories, which included good, medium, and bad. Good meant they were very similar, medium meant they were similar with slight difference, while bad meant they were completely different relationships. The sentence below was rated by good by the LM as judge, but the structure between for the subject of the Indra and the subject of our tool was were different because of the red things that Indra did not have, but they were very similar. So a use case for our tool was GraphRag. GraphRag stands for Graph Retrieval Augmented Generation, where we tried having the LLM to generate answers for a question. However, instead of just having the LLM generate a different LLM generate answers from scratch, we first retrieve a neighborhood of Bell statements around a specified gene set that we chose from a knowledge graph that was gotten by our tool that was stored in index. So this retrieved context, the retrieved neighborhood, is then inserted into a prompt alongside the question, how does metabolism affect DNA damage response? And then this was used to guide the LLM's response. So we tested GraphRag using knowledge graphs built from two papers by using our tool. One paper focused on cell cycle, the other paper focused on metabolism, and then we tested it on a single network and also made network from both publications. So like I said earlier, we tested it on the two papers. So at first, we told the LLM to answer the question without providing any network to it. And we noticed that the response gotten from the LLM was it gave a generic response, which was expected without linking to specific papers. When we tried giving the LLM context from a single paper network, which is the first cell cycle paper, we noticed that it mentioned genes that were present in the knowledge graph. It specifically told it that it should not make use of its background knowledge. So the next thing we tried was telling it to make use of its background knowledge with our knowledge graph. And we noticed that it still mentioned the genes present in our knowledge graph, but it talked about broader biology. So another thing we tried was merging the knowledge graphs from the two paper networks, and then we told the LLM to answer the question based on this merged knowledge graph. And it mentioned more genes and relationships from the both publications, and the answer was broader. So we also asked it to make use of his knowledge and the merged knowledge graph to answer the question. And we noticed that he explained more about the genes that were present inside our knowledge graph. So in conclusion, we developed a text knowledge graph tool that can be used to generate Bell knowledge graphs from literature, and it's available as a Python package, and you can also use it in Cytoscape web. Accurate entity recognition is still a challenge as this actually limits our output quality. We also realize that detailed prompt design and also giving structured output formats helped with the LLM responses, but entity recognition has to improve. We hope to make our framework adaptable to other formats aside from Bell in the future. Another thing that remains a challenge is the evaluation of the knowledge graphs gotten. This is because we don't have a gold standard to actually compare these Bell knowledge graphs to. Our project is available as a Python package. There's a link on the screen, and you can go to the GitHub repo to see how to make use of the tool. Also, the paper is up on bioRxiv. Special thanks to the Lunar Lab, the IDK Lab, Potato team, Indra team, and Google Summer of Code, where I participated in 2023 and 2024. Thank you so much.
Are there any questions for Favour?
Hey, hello, great talk.
Hi.
Thank you.
Thank you.
I just wanted to know if there is like a big advantage of using LLMs instead of like a relationship train NLP, like BioRex, I think, or something similar. Is there any big advantage of doing it?
The main advantage to why we used LLM was because of the like I mentioned, the fact that they are trained on a lot of biological medical information, and they can actually, there are some sentences that might have interaction that is not completely spelled out, but because we use an LLM, they will be able to understand that, oh, okay, this interaction exists in this sentence, even though it was not directly spelled out. And because of their like background knowledge due to being trained on a large scientific literature, they can bring out such interaction. And that is what happened when we tried Indra and the LLM. We had 67 interactions for 30 sentences, but just 29, because Indra just extracted like the ones that were clear, that you could clearly see, but LLM tend to like have understanding and extract something that was not directly clear or described in the sentence.
Okay, thank you.
Thanks. That was a great talk. So I'm really interested. I don't really have a question. So I'm just really interested in using this for the gene ontology, because even though we don't use Bell format, our GoCam pathway format can be mapped to Bell. And I just wanted to make you aware that I think, I don't know if Ido is in the room, but I think CAFA next year is going to have a competition for predicting GoCams. And I think this could be very useful to use for that, and that could
Thank
be.
you.
I encourage you to enter that competition.
Okay, thank you very much. So given that the tool is very generic, to adapt it to other formats will be easy. Why? Because the method is the same. So we basically just have to change the output format and give the structure and tell the LM, oh, we don't want Bell again, we want this, and like change some things. Thank you so much. Okay, there's one more question there.
I have a question. So how does the system handle conflicting evidence? For example, if one paper has a good relationship and other one has a conflicting evidence, so is there any, does the system has some sort of way to prioritize one evidence over the other?
Can you come again, please?
Sorry.
Can you come again? Can you come again?
Yeah. So is there any way to take an account to the conflicting evidence? For example, if one paper has one evidence supporting one hypothesis and another one has a conflicting evidence, so how does the system choose the correct results?
Oh, okay. So the thing is that you can process one paper at a time. So the system processes the paper, and then like for our Python package, it supports batch publication, but the system processes that paper before going to another paper. So it does not process it at the same time.
Another hand over here. Any other questions? Yep. One more.
Yeah.
Sorry, just building on that question.
Okay.
I think, is there any way that you filter from like bad papers, let's say, like just like something from a predatory journal, something like that? It's not like completely peer review. Is there any way that you can distinguish between a relationship like a gene with a biological process, for example, that has a lot of evidence, from one that just they say, oh, this is like that and that's it?
Oh no, currently there's no way to distinguish. Yes, we just get the interactions and the knowledge graph based on what you provide.
Okay.
Yes.
So let's thank Favour James again.
Thank you.